I will talk about 2 points. Okay. The first one will be bioinformatics, because as was shown by Jacqueline Chaffis just before, we need a lot of information to get more insights into the cell functioning, and we also need a lot of information to produce reasonable models of the cells. Of course, we are mostly interested in structural bioinformatics. So first of all, I will show you why we have entered an era where our data are called massive. So it's big data everywhere, and it's mostly now in biology. I will show you the numbers. Just to remind you, we came from a long history of trying to understand how the sequences are analyzed. And we started first to understand the laws of genetics. Then we were able to sequence in the mid-70s the first genomes, then the first genes, then the first genomes during the 90s. And from the beginning of the 21st century, we are now able to talk about not only one genome, but to a collection of genomes. So for one species, but also for many individuals in one species, and also for many different species. I will give you numbers. So you can have a better introduction of this data in the links provided here. And you can enlarge also the picture if you want the details. But the selling points are in 2001, the first human genome, the first draft of the human genome, and in 2005, the first complete human genome. So now we have genome, and we are willing during this this week to understand how these proteins interact each other to obtain the physical functions we are aware of. So this slide ends in 2003, and you will see that from this long history, the data available has very increased a lot. Because one major evolution happened in 2007. It was not representative enough of the whole genomes. So we have to account So from the cost of the first genome, which was nearly $100 million, it started to be much more cheaper, around $1 million in 2009. So it means that within less than 10 years, the cost was divided by 100, which is more than the Moore's law that you see here. And it fell dramatically to a cost which is now in 2018, so you see here the number from last year, less below 1,000 euros or dollars. So it means that for your own genome, you can buy it for the price, so you can get the whole sequence, the whole analysis of your own genome for less than the price of the latest smartphones. So you can decide in your life, do you want a smartphone or your genome? This is not the question we are answering in research, because of course we want to know what is the genome of each individual to know what could link to a disease or to another one. Just to give you numbers, this analysis gives for one genome more than 50 gigabytes of data, of raw data. So it's very big. If you take into account all the data available by now, it's already 100 petabytes. To give you numbers, because these are very huge numbers, if you compare to the Facebook production from the beginning of Facebook, it means 200 petabytes. So we're producing now half of Facebook of what Facebook has produced in its whole life. And YouTube is producing, so the major producing content in the world is producing nearly 400 petabytes per year. So it means that by now, we are filling up tons and tons of hard drives with biological data, and we need to assess what happens. The costs have decreased a lot, so we get a lot of data, we get a broader overview of what is a genome, and so we'll discuss how we can apply this to protein-protein interactions. If you want more links, again, I've provided some links about this and how to analyze this. So what happens now is, in any project, you can think of what you would get for one genome, but for the genome of the related species, for instance, monkeys, for humans, from Saccharomyces cerevisiae, you can get a lot of information from bacteria, from viruses, so you can link even species within each other. So it's never seen, and you can get a very big amount of information if you want to compare the interactions of proteins within the same genome, proteins which are autologs in other organisms, or the way viruses or bacteria interact with us. So here again, you can think about protein-protein interactions in terms of cancer, of infections, and of any disease you can think of. What happens now is, at the beginning, we were thinking that there will be a lot of proteins in our genome. It was assessed that we would get around 100,000 proteins in our genome, but now it's much less. We have around 20,000 genes, and this number is decreasing. But we have now much more information coming from other parts of the genome which are non-protein genes. And we know that this is very important for epigenetic modifications, the control of expression in different cells, etc. So if you think in terms of protein-protein interactions, why my protein will be expressed in one cell and why not in the other one, then you have to check if there is something that is linked to the protein you are looking at. The main problem for structure now, to link both information, is that the amount of sequences is really growing rapidly. You see from the graph here that the number is above now 110 million sequences of genes in the data banks. So it's a very huge number. But what is posing a real challenge is that by classical approaches, and I will just detail them a little bit afterwards, we only get about 140,000. So it means that less than one out of 1,000 is missing between both information. So we have a lot of sequences. We have a very few structure of proteins to support these sequences. So this is a very big gap between both information. And when you want to assess the structure of one protein binding to another one and with a lot of allostery in between, then you need to fill in the gap here, because most of the time you lack a part of information, of a lot of information. So we'll show you that it's not even there at the protein level, at the sequence level, but on every analysis you can think of now, you will get a massive amount of data. You will get, we are in a polyomics era. So I gave you the number about only the sequences, but you can think of the same amount of data on any project that you will find. Just to give you an example, it's a study from 2014 on wheat, and you'll see that on this project, they used a lot of approaches to get the details of epigenetic modifications, the influence of the soil, the influence of the interactions with the environment, with pathogens, etc. So you get a much broader overview at the organism level that we have never been able to do before. And so if it's done for wheat, it's done for everything. It's done now for so I mean it's very common. If you go to the hospital now, if you have a rare disease or if you are disease resistant to a treatment, they will sequence your genome. They will not give it on public databases, of course, but it's done every day. If you want another example, here is how you can think of what changes between an undifferentiated cell and a differentiated one. And you see all the factors here that are available. So here, so the factors. And the analysis that you will be able to access. So during this week, we will show you how you can access, even in Nantes, all of these parameters. And of course, you will get a lot of data to analyze. So what is worse, to give you the big picture, is that when you know the sequences, you know how many proteins you are supposed to get from these genes. So you know that on average, you will get about 10 to 20 proteins from the same gene. We are on eukaryotic genomes, so you get introns and exons, and you have combinations of them. So most of the time, what you will get, for instance, in viruses or archaea or bacteria, you will get more or less one protein or two proteins from the same gene. In eukaryota, it's much more complete, much more complex. It means that for the moment, considering all the data that I gave you, we know the structure of proteins for only 4% of all the proteins that we know are. For about half of them, we are in the gray region. It means that we don't have the complete protein, but we have more or less the part of the protein. So in that case, you can infer a lot of information from this. You can infer from other organisms. You can infer from various analyses. But still, there are dark regions and dark proteins for nearly half of the proteins. So it means that by now, we only know what are doing the proteins for, I mean, on the structural level, we only know half of all proteins are available. So it's a very big gap missing. And I will show you how using in silico approaches, you can try to complete this information. First, I will show you what is susceptible to work and what is susceptible to be problematic. So, to sum up, why do you need the protein structure? So I will focus mostly on proteins since we're talking about protein-protein interactions. First of all, we want to understand at the molecular level what can be in agreement with the experimental data or not, first of all. Second, which is very, very difficult to assess, is how the dynamic behavior can be analyzed or predicted. So like this, you can go to the bench and do something to assess that. What we are willing to do, and this is the main work for the next following years, is to assess how a single variation on one individual will have a consequence on size, on weight, on color skin, for instance, but also in cancer, in immune sensibility, etc. And of course, when you have understood these approaches, you want to design new drugs to block something specifically. So I think I've convinced you that it is important to understand from where we start, what we can do with it, and fortunately, if everything goes well, what we can do to fill in the gap between our knowledge and the I will now just give you a small reminder, probably a reminder for most of you, about what you can do with structure of macromolecules. So first of all, where will you get them? You will get them in the protein database. These data will come from NMR, from stats, from also some models. So you will get models produced by others, and you will get this information. We will have a talk also about the electronic microscopy, and there you will get valuable information on the organization of big structures, and it's very important for getting this information. And when you get no information or not enough information from experimental data, then you can on your own start to model using servers like the Protein Model Portal or the Swiss model. And as was already highlighted, there is a strong need for better proteins bound to the membrane, integral to the membrane. So it's very important in the following years. And of course, just to remind you, the first thing to work on a model, on a protein interactions, is to do a literature survey first. It's very important to know the protein you are working on before trying to use any of these tools. Otherwise, it's very useless. So, just to remind you, this is just a small introduction, but I gave you some links that you should start with. So if you go to the protein, you will get a broad information. You will get also some and will be very, very helpful for your own career. So just to remind you some of the experimental techniques. The first one is X-ray crystallography. The X-ray crystallography is application of your protein to a very high level. Then you have to crystallize it to get a crystal. Then you will get a diffraction pattern, and you will get the electron density from which you will get the atomic model. And you will have to refine. So this requires a lot of energy, of time, of money, and of knowledge. This is why this is very important if you want to have the details of atomic interactions in a protein. Meters, but this is just to sum up. This is the most precise technique for now if you want to get interactions of the structure of macromolecules and of proteins. If you want to get a more dynamic view of your macromolecule, you can use NMR techniques. So you will get the interaction. So this is only one card out of many cards that you will get from the interactions of atoms. And so you can get proper. Since you have inserted here, you will get a more dynamic view of your system. So you may have, like in the crystallography, very precise positions, and you will see broader zones where the interactions are less precise or more dynamic. So you will get an ensemble of conformations. So it's less precise than the crystallography, but more dynamic. So depending on your question, it's very interesting to get both. If you are looking at very big systems, then the emerging technique, and it was the Nobel Prize last year, so it's very recognized is you can use electronic microscopy to get the overall volume of your macromolecule, and then you can reconstruct it using X-ray structures of individual partners, the global shape. It's very important because it will get rid of many limitations of the two other techniques, because you can even go close to cellular systems. So you will get a lot of information tomorrow about that. But this is very developing a lot, and it will probably bring a lot of information that we will not able to gather before. When all these information are not sufficient, are not able to be used for answering your own questions, then you have to do molecular dynamics, molecular characterization, sorry. So in silico experiments, three different structures because I have no preference for all of them. Depending on the needs, you will probably need one or two. You will get a presentation on PyMol and on Maestro. I'm giving you links, and you have in your bags some documentation about that. And if you want to understand why it's important to use a lot of these approaches to integrate a lot of information, you can have a look at this small introduction, which is from the RCSB channels, from the PDB database channel, where you will see very interesting interactions and the the dynamics of the system. So these are not cartoons, these are the results of real simulations. So you should have a look at that, because this means that many months of calculations were done to obtain this image. So this is not only an image, it's the results of calculation. You have also to think into, you have again to take into account that a protein is never alone, as you will find in any lab experiment. So, of course, you have to analyze it and to cut the protein into smaller pieces. But never forget that the protein has to perform a lot of its steps before being produced, starting from the gene, which is transcripted, then transcribed. There are post-translational modifications on the protein, which are sometimes very important for the function, very important for the cell localization, very important for the recognition by other partners. So it's never natural, and you have to take this into account when you want to do a model. The second thing you have to think of is a cell is not void. It's far from being void. You have here an illustration from David Gilbert. You can go to this website. There's a lot of good explanations on molecular systems, and if you are lucky on your own molecular systems, there are very brilliant explanations, simplifications, and you will get a broad overview already within a short period of time. And to come back to this specific cartoon, you'll see that the content of a cell is very, very packed. So it means that you will never see a protein that, like you will see in your modeling, completely free, without any alteration, etc. It's very, very compact, so you have to take this into account. Next, I told you you have a lot of information to take into account. You have a lot of information, but we know there are a lot of missing steps, and the evolution of the techniques are not sufficient to cope with from the technique in structured determination is not sufficient to cope with the availability of all the other data. So you have to take into account the structural prediction when you model your protein. I will just detail you what you should use, when, and why. The first thing, so I told you we are lacking a lot of proteins, and these proteins are very difficult to study by statistical means. That's true, but we can also benefit from other factors that I will explain to you here. The first thing is the structure is 3 to 10 times more conserved than sequence. So even if you see a big difference in sequences, you can completely infer for some families of proteins that it will have the same shape, the same fold, the same organization. So it's quite easy to explain, because if you take any human on Earth, it will have a very big difference probably with your own genome, but it will still stand on his feet. He will have 2 arms, 2 legs, one head, and so you will be able to recognize it everywhere. So it's very easy to recognize. So the shape is much more conserved than the sequence, and up to 10 times more. So it means taking the opposite, that even on some proteins within a range of 15 to 25% of sequence identity. It means that if you look at the sequence, you will never find any corresponding amino acid, or very seldomly. So it will be very difficult. Even in these experiments, the proteins are very close in shape. The best example for that are the GPCR family, where the protein sequence have evolved a lot, but they all possess 7 transmembrane domains, and they nearly all look the same if you are not familiar with all the details. The second thing is also that there are a significant amount of subfolds, so of motifs that you will find derived from certain sequences or from their physical, chemical. So I've named you some of the folds because now we have a lot of them, but these ones are historical. for instance, the Rossmann fold or the Greek key here. Here you get a motif which is very common for DNAs, where you will get a zinc finger, you will get helix turn helix, which is very common in helices also, or helix turn helix here, which is very important for DNA recognition. You will get also hairpins like this, so one strand going up, the other strand going down. So these small motifs can be used as blocks for building proteins. So even if you don't have if you have no idea of the sequence, you can use these building blocks to start to build your protein upon. If you take this information into account, this is the overall procedure you will get in all of your analysis. So either the server you will use to get the information, or the control step that you will do on many different servers to get a proper model, a valid model. To the protein you want to model. Then you can define some motif, identify them on the structure, and then map the correspondence. And the simplest case, because in this situation you can completely transfer one information to another one. Okay, another thing is, even if it seems completely unphysical, it seems that the number of folds, so the number of shapes each protein can adopt, is not completely infinite. We are seeing a plateau since 2010 from the structure of the shape of new proteins. So the shape of a protein, the arrangement in 3 dimensions is called a fold. And we are seeing a decrease in new folds evolution since 2010, and you see here the total number of folds, and it's below 100 and 1,400. So it's big, but it means that any protein sequence that we know should be inserted inside of all these folds. So it's not that complicated if you take this problem like this. The other one is very obvious, but I want just to remind it because it will be used in, all throughout the rest of the week. Of course, we know the individual characteristics of each constituent, so I will just focus on amino acids. But we know all the amino acids, we know all the nucleic acids, the sugar, the carbohydrates, the lipids, etc., cofactors. Just again to remind you, we have all the amino acids. We know if they are charged or not, if they are aliphatic or not, if they are polar or not, if they are big or small. And you can even define Venn diagrams like this to isolate them. So this will not change depending on protein structure. They are individual blocks. We also know that there are many mechanisms which will lead to the protein structure, and we'll go from the protein sequence to secondary folds to the tertiary structure, and then sometimes you will get protein interactions, which are called quaternary structures. So we know that we will end up with something like this, and this is mostly thermodynamically driven. So it means that if you take any sequence and you put it on water, it will spontaneously fold into the native protein fold. So nature has optimized the protein fold. It will not produce something different than the one fold, otherwise you will get a disease. And another thing is we live in a world where we can put some interactions into equations. So I won't go into details, but just to have a small introduction because it will be presented more in details afterwards. But we can define all the interactions between atoms by terms involving bonds, angles, dihedrals, interactions between a lot of the van der Waals interactions and the electrostatics interactions. And of course, this is very interesting because as we are living in the same world everywhere, we can use the physical parameters, these equations, to have a reasonable description of the energy of the system. Now, after we have a lot of things that we can build upon. Then this leads to this information. Since we have a lot of information coming from different parts, and this is why I gave you a lot of information and a lot of places to look at, we can use the transitivity principle. So it means that if you know enough information for one protein and you can infer some information from other proteins, then you can have a better description of your protein or of your protein-protein interactions or of your network, because it is reasonable to use transitivity. It's possible to deduce some parameters because it's logical and it's solid. You can do that. Just to show you what we can infer, what is reasonable to infer, this is a very interesting example from the aspartyl tRNAs. You can recognize here the RNA which is fold, which has a T fold, and then you get here the bound protein, which is a small block here compared to the rest of the pictures. If you go throughout evolution, through time here, so you can see the distances between many different species, you can still recognize the RNA part. You can see what was gained during evolution and what evolved. So here from this small block, you will get, for instance, here, a small helix now building upon. Then new events appear, so you will have a small modification here, but a bigger one here. And then you get another part gained from evolution, and again another one. So if you look closely at the first part here, then it has not changed a lot during evolution. And we're talking about millions of years of evolution. So it has very big implications for structure, because if you don't know this protein, but you know this one, or this one, or this one, then you can infer, at least correctly, part of the structure for the rest. So you divide the problem into smaller problems. And as I gave you a threshold before for membrane proteins, we have said that for globular protein, the threshold will be around 30% of sequence identity. And just to show you what is sequence identity, it means that at some positions you will get exactly the same amino acids, and on other positions you will get something different. It means that even if we have a small amount of protein structures, then we are not that blocked, because taking into account all of this information, then we can predict the structure of many different proteins that are not known for now, and that maybe never known because of experimental limitations. So to sum up, what you should use in these situations, we'll define many different categories. I'll go back into each of them just after. But first, we have the most simple category, which is comparative or homology modeling. Comparative means that you will have something that you can compare of. It means that you will you have some evolutionary link between both. And homology, homology means you will have an evolutionary link between both proteins. Comparative means that you know that it should be the same. For instance, if you think of a pore at the membrane, you will have another. So what is important is the and not the structure of the detailed structure of the pore by itself. So you will need the function in that case, and so you may use comparative modeling for that. So if you have one protein and you have some discrepancies between the sequence, you see the threshold now is 40, it was 30 just before for the explanation. Then you have one structure, you can predict the structure of the other one using comparative or homology modeling. When you don't have the full structure, but you have some parts of the structure, then you are in the threading category. In that case, you will use subparts of the proteins to assemble them to produce the complete protein. So in that case, like using small parts of them, the difficult part lies in the fact that you will have common structures that you will have to match to one structure. So you will have to decide, are you taking only this helix or the helix plus the beta sheet here to produce this one, or use this beta sheet for this one? So of course, it's much more complicated than the first category. And the last one is when you have no luck. And unfortunately, I told you it's half of our proteins from the very big amount of proteins. We are lying in the ab initio category. So in that case, you have the sequence and you want the protein structure, but you have no idea of how to transfer it. So it's the most complicated thing to do. And of course, it means that if you take the fiability index in red here into account, it means that here you can nearly assess that this protein, the model of this protein is correct because you have a fiability index of 80, 90%. If you are here below 25%, you're nearly sure that at least part of the model will be problematic or even completely wrong. So, depending on your needs, before going to protein-protein interactions, if you want the structural details, you will have to produce part of your protein, all of the protein, all of your interactions. So what you will need in that case is using different methods. I will just give you examples of some methods afterwards, but since it's a very important research field, there is an international competition which is called Critical Assessment of Protein Structure Prediction, CASP, and you can get the live results here. There's an ongoing competition this year. I think we'll get the results by the end of the year. So what I'm giving you for the moment may change depending on the results. So you have to check this very regularly. First of all, I will just give you a small introduction of what you can do for homology modeling. The main principle that you will see most, in many different approaches is from your protein sequence, you will search which protein are available, for instance, for serving as support of your analysis. So you will first browse the protein data bank. I told you that's where all the proteins are stored. So from your own sequence, you will see if any protein known is matching something. And then you can see even on a multiple sequence alignment the results. If your protein sequence is covered completely or half or sparsely. From this, you get a profile. And you can use, for instance, an objective function which is defined in the software modeler, which is one of the best in this category. You will tell him which templates you want to use for producing your model. You will use, it will use a lot of constraints defined internally, like stereochemistry, physical chemistry parameters. I reminded you the structure of amino acids. Here you have a lot of parameters taken into account. It will sum all of these constraints into an objective function, optimize them, and then it will produce the structure of your protein, of your macromolecule, using all the constraints that you give. Never forget, it is a model, so it's not the definitive step, but the first step to do something else afterwards. Because the software is available, there are websites also to do it automatically, but it's really the first step that you have to take into account. Next, so if you just want to have a first site at your protein, you can use their own ModWeb server, and it's only one out of many. You can also use Swiss model, which will help you to produce your model in more steps. So these are online tools, so you can provide your sequence and you will be able to define your own steps or let the tool work automatically for you. But for this category, I told you it's quite easy. So you can more or less trust the tools and just arrange the model at the margin, and you will get a reliable model that you can build experiences on. When you're in the less reliable category, which is called threading or enfilage en français, then you will need more information, you will need more control to assess the model. In that case, the protein will be defined as an assemblage, part of many different substructures. So if you are lucky enough, you will get enough substructures that you will just need to assemble. So it's a complicated problem, but not too much and not using the computing power for now. So the first thing is to find which are the structures. So like the first profile that we had before, you will define which structures are close to the template. Then you will assemble them using profiles and then refinement steps. And in the end, you will get a model that you will be able to use. It will produce a PDB file that you can look at. And it will be up to you to decide if the model is precise enough or not. So in that case, you will have to remember that in this situation, you will have to go again to the literature. Is my model in agreement with known data, with binding data, etc., interaction data, or not? So it's on your own. In the last, so the most reliable throughout many CASP experiments is iTaser. So depending on the years, it's not always the top performing model, but in the on average, it's very, very good at producing. It will do all the steps for you. It's just summed up here. You can find it again on their website. You will take your own sequence. It will do a profile, try to find low resolution templates, sorry, and then assemble them, cluster them to get different solutions of your problem, then refine them again, find the best solutions, and you can even define some parameters, because based on the structure it's possible to derive some information, like is it an enzyme or not, a binding site is present or not, etc., etc. So this method is quite robust, but depending on your needs, if you want to see a precise interactions between two proteins, you will need more work to attain this goal. So again, you will get a web server. You just register and you will launch your job. So this is the first step I'm doing when people come to me to say, is it possible to model my protein? Let's start by this, because it's very efficient at finding templates and at producing reliable models within a defined period. So you should always try iTaser. This is my advice. You do it. You do like you want. But it's very good for starting a project when you have no idea about the protein structure. And of course, that no protein structure is available. If you lie in the last category, then you are in trouble, because I told you it's the less reliable category. In that case, you will use two different approaches, de novo or ab initio. So there are subtle differences between both, but I will not define them here. In that case, you will see the proteins as a short assembly of small protein blocks. So these blocks will be defined in fragments. So you will take the whole PDB and split every protein in block of 3 amino acids or 9 amino acids, and you will need to combine them using many different rules, including a force field, the amino acid characteristics, different functions derived from other parameters, and you will get a global score of adequation between your model and what is known for now. So this is more difficult. I would recommend that this is the overall acceptation community for proteins which are very small, so below about 100 amino acids now. It has been proven that for many proteins which are below these thresholds, you can produce a reliable model from any protein sequence which is below 100. Above that, you are in the universe. You don't know where you go. The best performing tool for that, for now, is Rosita. Rosita will take any protein, define it as small blocks, assemble them. So you will start with a completely straight proteins. Then it will try to fold independently many different parts. Then it will try to refine it. If you are lucky enough, what will happen is from this straight protein sequence, nearly the same as if you were in the linear protein sequence, you will attain a reasonable structure, which you will find here, and you will be able to decide which model is good and which model is not good, because the energy landscape of the Rosetta function will just go sharply decrease and attain a minimum here. And we know that we are able to go there, which is called the funnel of energy. Then we'll go close to the stable form. Maybe not the complete stable form, but very close to. So in that case, you know the protein structure is interesting, and so you will be more confident about using this protein structure. Again, if you want to do that without controlling any parameter, you can refer to the Rosetta web server, which is called ROBETA. You can also use many different services which are important for protein interactions, like interface alanine scanning, like libraries if you want to produce your own models, domain parsing, etc. So many different tools. So it's very important. You should use if you want to produce models. And of course, why I'm telling you that is that you can get a lot of expertise on all these approaches by doing it yourself. And it's even better than doing it yourself using the website, because Becker and his colleagues have produced a folded application where you will get a lot of training from the application itself, because it's real science. So you will get much more training what I have the time to show you now. And just to tell you, this is not playing games. It's playing it's science with games, which is very interesting also. Some of the protein structures that were used within Foldit were refined by the community, by a big group of users, of online players, and they were able to produce a better model than the crystallographic. So they were able to refine the crystallographic structure. And just to tell you, it's not a simple example. It was published in Nature, so if you don't trust me, read the paper. But it's really recognized by the community. To add another point is, if you take into account now back to the information that I gave you at the beginning, we have a lot of sequence informations. We have a lot of sequence coming from many different organisms. We know that we can transfer some parameters from one species to the other one, from one protein parameter to another protein. So we can infer a lot of information. And we have a lot of information. So if you are smart enough, if you have computing power, if you have broad expertise, then you can do what they did last time it was in 2016. So the team of David Baker did a big so I'm not sure it's very easy to read here, but you have the links to the papers here and the comment here. In their work, I told you there are 1,400 folds known. And half of the proteins, we don't have any idea of what the protein structure is. In their work, they were able to produce the structure of about 600 proteins. Out of these 600 proteins, 140 were not known in the PDB. So it means they have been able to decipher 140 folds never seen before. So it means that they have increased our knowledge by 10% using their bioinformatics approach. How they did that? They used all the parameters I gave you, so in a much more complex form also, and they used sequence analysis from a lot of metagenomic data. So metagenomic data is, you don't know for many species which species they are, where they live from, etc., but you can use the sequence because it's cheap. So you can say if something is important enough, if one amino acid is important or not, at which position, and assembling all of this information, you can produce new models. So we'll talk with Chris, reliable or not, but he has proven that for membrane proteins, for new folds, or even it was real. So it means that some crystallographer here had to change their mind or to change their objective because the protein was now defined. So it's a new way of seeing that. Of course, it's one shot. We'll see if it's possible to do it all the time. But just to tell you, it's not a toy science. It's very important. And if you want to study protein-protein interactions, depending on the needs of your experiments, then these approaches are available to all of us. So you have to gain expertise on using Rosetta, on using metagenomic data, but a lot of data is available, so you can benefit from it. And just to finish, because I don't want to be too long, if you want to go back into details into all the UniProt annotations, the structure, I gave you some links from the PDB. You should have a look at UniProt also, because there are many information on it, and it's very important to use these videos. They are very well. And if you want to have more precise links for trying to understand if a mutation is important, you can go to some services like Polyfen, like Pop Music. If you want to assess globally the conservation of amino acids, use web services like Conserve. They are available. They produce a lot of information that are very useful, especially for protein-protein interactions, because you can see if some hotspots if some amino acids are very conserved, and you can define all the parameters. There may be hotspots. You can get a lot of services from the Swiss or from the European Bioinformatics Institute. There's a website here which is free if you have some questions, some requests on it, which will answer most of your needs, because if you need a tool for predicting something, you can go there. And I think it's enough for today.